Hi, this is Michael Edwards, Assistant Professor of Medicine at UC Denver, specializing in bioinformatics. This is part 2 of the IPA tutorial. Was done in conjunction with the AACR workshop held in Snowmass in 2014. Our first tutorial, what we did was download our gene list. This was a list of genes different between two different forms of B-cell lymphoma, one more aggressive than the other. So in this tutorial, we're going to talk about how do we explore this list for pathways. Um, so when you double click on your analysis, this window will come up. It'll go to this first tab, which is your summary tab. And all this does is summarize some of the information that's given in all of these different analyses. Uh, so we're going to talk about the pathways right now. So I'm going to click on this second tab. Given here, listed by the odds of it, I'm going to make this a little easier to see. Given here are pathways with members overrepresented in our gene list, and this is sorted statistically. So the less likely you would see all these genes in these gene lists, the higher up on the list. Given on this scale here is the log p-value. So this would be the log negative log p-value. The higher this number, the more significant the result of this pathway being in our analysis. We can highlight over this. This will give you actually the actual p-value. So the odds of getting these 16 molecules associated with T helper cell differentiation is 1 times 10 to the negative ninth. This line here, and then this yellow line is the threshold. So about 1.3 is equivalent to a 0.05 p-value. So anything above this we can consider statistically significant, and some of these are way above. This yellow line represents the ratio. This is the amount of genes that we got in our list divided by the total amount of genes associated with this pathway. So in our gene list, we have 16 out of 71 known to be associated with this pathway. And we can click on these. Given down here are the genes in our gene list with its associated p-value and log ratio based on, and these are all represented based on the more aggressive B-cell type. And so they're all given down here. We can open up this pathway and take a look at some of these genes. Zero in here. In the paper that was associated with the data set, they mentioned that the aggressive lymphoma had a higher STAT3, and they believe this was one of the genes that was associated with it being more aggressive. Any gene or genes that are colored were on our gene list. Genes that are red had a higher expression in our aggressive B cell types, and those in green a lower expression in the same cell type. Close this window here. As you can imagine, if we just take a look at that, we can see a lot of immunotype pathways. And that's what you find, is that a lot of these pathways, even though they show up significant, share many of the same genes. You can go to the second tab, still under canonical pathways, overlapping. And this will give you an idea of some of the overlap on some of these genes. So if there's a line connecting two pathways, that means there's common genes in both pathways. We can filter this a little bit, make it a little easier to see. I click on that filter up there. I want to display 20 pathways, say, given here is 25. And in order for it to draw a line, I want at least 3 genes in common. I can hit apply. And now we can see that it's much different. You can see that most of the genes or the pathways here, and we can show the number of common genes, and we can show the p-values for the significance of each of these pathways. As I was saying, you can notice most of these are immune type pathways, and it's not unheard of for them to share many of the same genes. You can see some of these, though, out on the side, a little different. Hepatic fibrosis, hepatic stellate cell activation, these are more growth factor pathways, and we could take a look at that. But again, as the original data set talked about STAT3. So let's look at this. So the odds of getting these genes associated, we can click on that. So down below are all the genes in our gene list associated with this STAT3 pathway. Again, given the p-value and log ratio. We can click on any of these genes. Actually, the first gene on this list is B-cell lymphoma 2. Obviously, that makes sense compared to what we're looking at. We can click on any of these genes and get more information on them. Run. So this will give you functions, subcellular location, some of the pathways it's in, what regulates it, what is it regulated by, what it binds, roles in cell, and sometimes it'll give you a summary and also chromosomal location. Oh, there's a summary right down here. Get out of this. So again, we wanted to look at the STAT pathway. We can look at these genes a little closer. I'll double click on that. So here is the STAT pathway, the canonical STAT pathway. All genes in our gene list are colored. According to this, they're all red, meaning that they're going up. We can overlay our expression data on there. So I just went here under analysis. So first I go to overlay. I click that one. Then I select analysis and lists. And then we could just put our log ratio or p-value down there. We could also lay any other gene list that we come up with, and this is incredibly useful if we're comparing two different data sets. So here's our genes. You can notice some of these do not have p-values or fold change under them. These with double lines represent complexes, so there are many different genes are associated with this particular classification. We can look at those. So I select it. And again, we can put these anywhere we want. So I'm going to select it. I'm going to right click and I'm going to say show members. So given here are all the genes in our gene list associated with this particular classification that were different. And say we want to keep these, but we want to get rid of these, I can select those. I basically just drag a box around them, and there's a delete up here, delete selected. And now we only have those genes that we're interested in. We can also overlay potential directions of expression. So again, I hit the overlay. We can hit molecular activity predictor and start prediction. So now what it's done, the software has done, is it's coloring based on whether the software thinks it's more active or more inhibited based on the changing expression of the pathways that, of the genes that we originally imported. So blue would be more inhibited, orange would be more active. We can overlay all kinds of other stuff. We can look for drugs. So what the software does is find genes that are in this particular pathway that are targets of all the drugs that are out there. This one here has 3 targets. We can click on that. And now it's here. And if I select it, all genes known to be inhibited by this particular drug are highlighted in light blue. And you can see those right here. Some of the other functions, diseases and functions. It's coming. And again, you can see now that we're looking at this pathway that this is stimulating things such as tumor metastasis, angiogenesis, immune evasion, things that you would associate with a more aggressive tumor. So this is making sense, basically. You can look here and look at any of these categories for different genes. There's 19 genes associated with cellular developmental leukocytes, which you would obviously expect. Lymphocytes here. This pathway makes sense as far as being important in our system. Again, some of these others, and I'll go over some of them when we talk about some of the other tabs. But these are some of the tools that you can use, and these you can actually take this pathway and hit Path Designer up here. Yes. And now this gives you a more manipulative pathway, and you can make things bold. You can increase the font. You can increase the icons. You can add a legend. They're all up here. Um, so basically this is talking about the pathways. The next tutorial that I'm going to talk that we're going to do is going to look at biological functions and diseases that might be overrepresented in your data set, and how can we use this tool to get us an idea of what are the genes doing in our system, and potential ways to manipulate them. All right, thank you very much.